specifically yung fluorescent group po. Alright, so let's jump right ahead to Pseudomonas fluorescens and putida. So ayun po, Pseudomonas fluorescens and putida are both of very low virulence and they rarely cause clinical diseases. So they have been isolated from respiratory specimens, contaminated blood products, urine, even cosmetics. There have been cases of contaminated cosmetic products kung saan na-isolate po yung Pseudomonas fluorescens and putida, and also they have been isolated from hospital equipment and fluids. So Pseudomonas fluorescens and putida have been documented, although quite rarely, in cases of UTIs, post-surgical abscesses, empyema, septic arthritis, and other wound infections. So they're basically everywhere. So wala po silang specific site where they could cause an infection. So they could cause anywhere kung saan hindi nila usual habitat yon. Specifically for Pseudomonas putida, this has been associated with catheter-related sepsis in patients with cancer. Meanwhile, yung Pseudomonas fluorescens naman po, the isolation of Pseudomonas fluorescens in blood culture bottles from asymptomatic patients has been responsible for reported cases of pseudobacteremia, probably related to contaminated catheters and catheter-related devices. So yung Pseudomonas, why did it move? So yung Pseudomonas putida daw po, or fluorescens, sorry, yung Pseudomonas fluorescens daw po, it usually, why does it move on its own? It usually causes, ano daw po, pseudobacteremia. So usually, it could mislead us to thinking na yun nga po, the patient is bacteremic. Pero in fact, hindi naman po pala, and contaminated lang ito with Pseudomonas fluorescens. Alright, so for today's discussion po, we will be focusing mainly on Pseudomonas aeruginosa. Alright, so Pseudomonas aeruginosa, this has been commonly known as the agent of blue pus. So, marami pong iba't ibang pigments na pinoproduce ang Pseudomonas, pero ang pinaka-characteristic niya talaga is its blue-green pigment. So, Pseudomonas aeruginosa can grow at 42 degrees Celsius. And this would remind us of what other organism? Yung Campylobacter, di ba po? So, for Campylobacter, ang optimum growth temp nila ay at 42 degrees Celsius. And for Pseudomonas naman po, kaya din nila na mag-grow at this temperature. So, this makes them special kasi most colon microbiota or most commensals of the gastrointestinal tract can no longer grow at temperatures this high. The Pseudomonas aeruginosa is a non-lactose fermenter. So, expect na po natin na yung magiging itsura niya on McConkey ay clear colonies lang po. Pseudomonas aeruginosa can produce a variety of factors that could lead to its pathogenicity, or it could produce many various virulence factors, including endotoxins such as their lipopolysaccharides, their pili, capsule, or flagella. Do take note, though, that not all strains or isolates of Pseudomonas aeruginosa are encapsulated. Next, they also produce several exotoxins, which includes proteases, hemolysins, lecithinase, elastase, and DNase. And so the most important out of all of these exotoxins is exotoxin A. Alright, so yung exotoxin A po, this functions primarily, or this functions similarly to diphtheria toxin by blocking protein synthesis. Alright, now, some of them may or may not have capsules. I think I've already mentioned this. Hindi po lahat sila are encapsulated. So these photos on the right shows us the difference in colonies for encapsulated strains versus non-encapsulated strains. So yung mga non-encapsulated po, they would appear non-mucoid, just like this one on the top right. Tapos yung mga mucoid colonies po, expect natin na ito yung mga capsule formers. Alright, so marami pa pong modes of transmission ang Pseudomonas, and isa na sa most common is through the use of contaminated medical devices. So, one story that our former professor shared before was that there was this one time one of his relatives were confined in a hospital due to other reasons that or due to a disease that was non-infectious. Okay, so na-confine daw po or na-admit sa hospital yung relative niya um, because of other reasons. Tapos, after nung procedure na ginawa dun sa patient, a few days later, supposedly during the recovery period, napansin po niya na merong blue-green discharge na lumalabas dun sa catheter na naka-insert dun sa patient. So, pinaculture nila yon. And ayun nga po, na-identify as Pseudomonas aeruginosa yung isolate. So, imbes na towards recovery na yung pasyente, na-extend pa po yung confinement niya dahil lang contaminated yung catheters na ginamit dun sa patient. Another commonly observed route of transmission is through the use of homemade or contaminated contact lens solutions. So yung itsura po nun is same as the first photo that I showed you earlier. So it's this one. Ayan po. And another commonly observed mode of transmission for Pseudomonas aeruginosa is through swimming in unclean or shared hot tubs and jacuzzis, which brings us to one of its most clinically known presentation, known as the jacuzzi syndrome or hot tub folliculitis. So as we can see here on this photo to the left, this is an example of a patient with infected hair follicles caused by Pseudomonas aeruginosa. Another is the swimmer's ear. So same lang naman din po sila ng mode of transmission ng hot tub folliculitis. Alright, so di ba po nabanggit natin kanina that Pseudomonas aeruginosa can survive and grow at temperatures reaching 42 degrees Celsius, di ba po? So factor yun. Kaya naging common isolate ito among people who swim in these recreational bodies of water. All right, let's now look into the different pigments that may be produced by Pseudomonas aeruginosa. Okay, so first is pyoverdin. So pyoverdin, this is responsible for the yellow or yellow-green appearance of some colonies, as we can see here on this first plate. Yung pyocyanin naman po, this is the one that is responsible for the characteristic blue or blue-green colonies and also the blue-green pus that is observed during the course of infection. All right, as you can see here in this plate in the middle. So ito pong itsura ng pyocyanin. Next naman po is pyorubin, as we can see here on the third plate. Ayan, this is responsible for the pigmentation or the red pigmentation of colonies. So that was for pyorubin. And lastly, pyomelanin po, if the bacterial strain happens to produce pyomelanin, they will appear to be brown or black. Take note though na special po ang pyocyanin dahil Pseudomonas aeruginosa lang po ang kayang mag-produce nito. Alright, so lahat po ng ibang pigments na to, kaya rin silang i-produce ng iba pang species ng Pseudomonas. Pero po, yung pyocyanin, si aeruginosa lang po ang meron yan. So please take note of that. And here is a list of tests kung saan po magpa-positive ang Pseudomonas aeruginosa. Take note of the usage of the cetrimide agar. So the cetrimide agar po, this is a selective and differential medium that enhances the production of pyocyanin and pyoverdin. So ito pong picture dito, most likely ang ginamit po dito is the cetrimide agar. So although it was originally formulated to enhance the production of pyocyanin and pyoverdin, ma-observe pa rin naman po natin dito yung iba pang mga pigments nila. And then next, colony morphology on BAP or SBA. Pseudomonas aeruginosa has been known to produce colonies with a characteristic metallic sheen, as we can see right here in this photo. Alright, so kung i-compare po natin siya sa E. coli, so di ba po yung Escherichia coli, it will produce greenish metallic sheen on the eosine methylene blue agar. Pero po kung BAP ang ginagamit natin and we see that the colonies were able to produce a metallic sheen, most likely po the isolate is Pseudomonas aeruginosa. Let's now talk about the other miscellaneous species of Pseudomonas, specifically those that belong to the non-fluorescent group. So first up is Pseudomonas stutzeri. So Pseudomonas stutzeri, although a rare isolate and even a much rare pathogen in the clinical laboratory, it is usually easy to recognize because of its characteristic morphology or characteristic colony morphology. So ayan po. It has been described to be wrinkled, leathery with a light yellow or brown pigmentation. Alright, so Pseudomonas stutzeri in immunocompromised host has been reported to be responsible for diseases that include septicemia, meningitis, pneumonia, especially in cystic fibrosis patients, and also endocarditis, post-surgical wound infections, septic arthritis, conjunctivitis, and UTIs. So basically, they could attack anywhere if the patient is immunocompromised or if they have HIV. Next is Pseudomonas mendocina. So this can be found in soil and water, but it is rarely isolated from human specimens. All right. So when it is actually isolated from human specimens, it is often considered as a contaminant. But of course, there have been rare cases of endocarditis that has been documented kung saan po na-isolate nila yung Pseudomonas mendocina. So Pseudomonas mendocina produces non-wrinkled, flat colonies that may appear yellowish-brown in pigment. So in terms of pigmentation, magkahawig po yung Pseudomonas stutzeri versus yung mendocina, but they would vary in texture. So Pseudomonas stutzeri is wrinkled and leathery, while Pseudomonas mendocina is non-wrinkled. Next are Pseudomonas alkaligenes and pseudoalkaligenes. So these are often considered as contaminants when isolated from clinical specimens. So they are oxidase positive, and for the rest, they are biochemically negative in many of the tests for which Pseudomonas species would usually test positive. Alright, so di ba po kanina nakakita ng listahan ng mga tests kung saan magpo-positive yung Pseudomonas aeruginosa. So if we were to compare Pseudomonas aeruginosa with alkaligenes and pseudoalkaligenes, magpo-positive sila for oxidase but everything else negative na po. They are also motile by means of a polar flagella. All right, and then last ones are Pseudomonas luteola and oryzihapitans. So the natural habitat of Pseudomonas luteola and oryzihapitans is actually unknown, although both species have been isolated from soil and water. So Pseudomonas oryzihapitans specifically has also been isolated from hospital drains and respiratory therapy equipment. So these two species are rarely isolated from humans, but they have been documented in cases of wound infections, abscesses, blood cultures, even peritoneal fluids, and they have also been implicated in cases of peritonitis and possibly meningitis. Although this only happens often in association with other bacteria. All right, so usually daw po Pseudomonas luteola and oryzihapitans infections by themselves. Rather, they work together or kind of like in conjunction with other bacteria. All right, so now we're done with Pseudomonas species. Let's now proceed to Burkholderia species. So all of them are motile except for Burkholderia mallee. So there are 4 species in this genus, but ang focus lang po natin for today is Burkholderia sepacia, mallee, and pseudomallee. Alright, so starting off with Burkholderia sepacia, ito lang po yung mga kailangan nating tandaan dito. So this is the most clinically documented cause of bacterial pneumonia among cystic fibrosis. Alright, so kapag nabanggit po na bacterial pneumonia among cystic fibrosis patients, dapat po maisip nyo na agad is Burkholderia sepacia. And ito po yung mga morphology nila. They are the help of the B. cepacia selective agar. And either if we use the B. cepacia selective agar or on a routine BAP or SBA lang po, same lang din naman po yung magiging appearance nila. So they have been described to be smooth with a yellow or yellow-green color, as we can see right here in this photo. Next is Burkholderia mallee. So this is the causative agent of glanders, which is a disease. But for humans, this can cause acute pulmonary infections. This has also been classified as a Category B bioterrorism agent by the Centers for Disease Control and Prevention. Alright, so nabanggit ko rin po kanina sa general characteristics that Burkholderia mallee is the only non-motile species. Next is Burkholderia pseudomallee. So commonly known as the Vietnamese time bomb, this is the causative agent of melioidosis, which have been described as a glanders-like disease. So magkahawig daw po yung clinical manifestations ng glanders and melioidosis. And ito po, listed here are the possible clinical manifestations of the patient. And of course, since respiratory tract infection po or respiratory yung clinical manifestations ng pasyente, the mode of transmission po via the inhalation of bacterial cells. Other possible modes of transmission include ingestion or inoculation of the organisms. So with regards to its epidemiology, they can be isolated along the soil or muddy lands of Southeast Asia, specifically sa Vietnam. So very abundant po yung bacteria na to along Vietnam, which is why it has been given the nickname as the Vietnamese time bomb. Alright, so tandaan po yung unique coloring morphology nila, which has been described as dry and wrinkled, as we can see right here in these photos. Alright, so tapos na po tayo with the Burkholderia species. Let's now look at the other miscellaneous gram-negative bacilli. Okay, so right here we have Stenotrophomaltophilia, and kasama din po ito dun sa mga bacteria na kayang mag-grow at 42 degrees Celsius. Also, as its name implies, Stenotrophomonas maltophilia, it is a maltose fermenter. Alright, so madali lang po natin siyang ma-identify if we were to run it through the carbohydrate utilization test. Again, this would give us fermentation. Alright, so Stenotrophomonas maltophilia is a common contaminant of blood drawing equipment. So, although wala po siyang tunay na clinical significance, kailangan pa rin po natin siyang aralin because we have to learn the differences between actual pathogenic bacteria and yung mga contaminants lang pala. Alright? So, ayan po. Now, on blood culture plates, Stenotrophomonas maltophilia would have either a lavender green or light purple pigmentation, as we can see right here in these photos. So ganito daw po yung itsura if it produces somewhere around a lavender green pigment, and ganito daw po yung itsura if purple yung pigmentation nila. Alright, next is chromobacterium violacea, and as its name implies, no? So this produces a violet pigment known as violacine. So included din po siya sa samahan ng mga bacteria na kayang mag-grow at 42 degrees Celsius. And again, for most enteric or colon bacteria, inhibitory na po yung ganito kataas na temperature. Alright, and mag-isa lang po siya sa genus na to, so wala pong ibang species ng chromobacterium maliban sa violacea. And this is only an opportunistic pathogen, and it is very rarely encountered in actual practice. So again, inaaral lang po natin sila to be familiar with some of the organisms that may appear as contaminants in clinical samples. All right. But of course, as an opportunistic pathogen, it may sometimes cause neutrophil deficiency in immunocompromised patients. So usually ganun naman po, di ba? Makakapag-observe lang po tayo ng, or may only link them to actual clinical significance if the patient is immunocompromised. So sometimes we have to make those correlations before assuming kung yung na-isolate nga po ba natin is the actual pathogenic bacteria or kung ito po ba ay contaminant lang. Alright, so here is what chromobacterium violacea looks like on culture. Alright, so here are the references that were made or that we used in the making of this discussion, and ayan po, thank you so much po for listening. Our chat box is now open for questions.